So let's go and get started. We're talking about phylogenetics. The last piece that we talked about was the fact that you can use behavioral traits to make a phylogeny, but it's kind of silly. Over the weekend, you read an exception to that where there was a conflict between the morphologic and the genetic phylogenies, the two most common type of trees or traits that are used to create trees. And they used, that was actually pretty damn clever, different type of nesting strategies, different types of socialization strategies in order to evaluate an agreement between these two traits. Someone can please remind me, why don't we use behavioral traits to make phylogenies? Why is it basically just impractical? Just isn't that many behaviors. And if you look at like a tree like this, the number of hypotheses that compare the relationship between its different species in this phylogeny of leafcutter ants, billions is a small estimate. Billion alternative hypotheses is a small estimate for that size tree. You don't have that many traits to deal with it. So you got to come up with different strategies. One, you have to have a trait that is hyper common, and then you have to use different statistical procedures like heuristic analyses and bootstrapping in order to resolve on a tree. So behavior just doesn't make any sense. It can work because it is a trait that evolves via natural selection, therefore it's inherited, but it just doesn't make any sense. Instead, what we do, much like we did in this tree, is we character map. Well, we are way, way off. Character map. You know, determine the evolutionary history. How many times something evolved? The evolution of a complex trait. What were the steps of that trait? And we apply these to behavior very often. Sort of a simple example that kind of builds off of our previous one is, how often did sky pointing wing waving evolve? And we have two basic hypotheses for these. We have one situation where it evolves, and then it is lost 5 separate times. So it evolves here, which is why we see it in this group and this group, the behavior we see sky pointing wing waving. But then it's lost within this clade, it's lost within the hingas, it's lost within this group, and it's lost within this group. So it emerges once, drops out 5 independent times. That's one hypothesis. We use a tree that's known, and then we place the characters on top of it. Another possibility is that we only see it. So I have these two backwards. One possibility is it evolves twice, period, independently, in this cluster and this cluster. The second possibility is that it evolves once and is dropped out 5 times. Now, when we character map and we put our traits onto these trees, we use an assumption. And we talked about it before. Someone remind me what that assumption is, which is up on the screen too. Starts with a P. It's going to be that morning, isn't it? Parsimony, thank you. The parsimony makes sense. It's a good assumption. The fewest number of steps, the least complicated pathway, the least complicated evolutionary tree is the most likely one. And that's a very reasonable assumption. Is it an absolute? Is parsimony always going to be the correct tree or the correct interpretation? The answer is no. No, and we have improved in sort of our statistical modeling of how to accommodate that based on the likelihood of something emerging versus something being dropped, especially when we understand some of the underlying genetics. But it's still a very good assumption. It's kind of like an adaptive paradigm. It tends to work in the vast majority of cases. But as with an adaptationist paradigm, we always do want to be a little bit cautious. recognizing that there can be alternatives in terms of fully understanding the overall system. Now, using this idea of character mapping, we can examine some really profound and weird behaviors. And one of them is something called brood parasitism. Let me see if I can get this going up. So you saw the host bird in the beginning, the rufous collared sparrow, laying its eggs. You see the presence of the eggs. This is the brood parasite. And what they do is they lay their eggs in a host nest. So they don't actually provide the parental care. They essentially lay their eggs and expect the other parents to care for their offspring. Almost always a different species. And what this paper is looking at is how the egg laying process of the brood parasite damages the host eggs. So the way they drop those eggs in actually causes the host eggs to crack and therefore the host chicks to die. These are host parents trying to chase off the brood parasite, but without success in this case. So there's a few things that's kind of cool about this system. One, the brood parasite, these cowbirds in the Americas, cuckoos in Eurasia, they get the parents of different species to adopt and care for their offspring. And they can do this through a variety of mechanisms. One of the things that you see here is this egg is being laid in. That's a brood parasite relative to the host nest. One of the things that brood parasites will frequently do is they will remove one egg for each egg that they lay, with the idea that some species of host parents can recognize the number of eggs. And if there is a wrong number of eggs, those parents will abandon. Frequently, the individuals that get parasitized are first-time parents. Further, when the brood emerges, the brood parasite emerges, they tend to be much larger and they will kick out their adopted siblings, killing them. So they'll just basically push up their back, push them right off the nest, and therefore being the only individual in there. And we'll see later in the semester that they can actually use signals that get the parents to feed them more by mimicking not the call of a chick, but mimicking the call of an entirely full nest of chicks. So they can make themselves sound like more than one individual. And as I got to walk around with a buddy yesterday, I've had 3 children, but all evenly spread out. One, 3 years apart, all born in the exact same week. We have a mating season in my family. This buddy had twins, and you could just see how much more work the twins were because one twin would go this way, and they're about 2. And the other twin would go this way, and there's only one parent there to try to figure out which one to catch at this time. One would lay down on the ground, start to lick the asphalt, where the other one would go into the debris and start grabbing up all the leaves, putting them in the mouth. So you could just see where like the amount of activity causes that much more work. So they do all these things. So one of the things we see in North America is we have different species of cowbirds. And what you see on top is the number of host species that they can parasitize, all the way from Rufoloxarius, which does 1, to adder, which can do up to 216. Is anyone familiar with Van Valen's Red Queen hypothesis? Can someone give me Van Valen's Red Queen hypothesis? There are favorite books in science. If you know your Victorian literature, you know the Red Queen from Alice in Wonderland. And her statement about staying in place. For every step forward we do, we go 2 steps back, and basically just running in a big circle. And Van Valen was a theoretician from Chicago who made an argument that evolution was prone to specialization. And the reason it's prone to specialization is because of interspecies competition and intraspecies competition. Competition among individuals of different species. And then once that's resolved, competition within a species. And it makes a lot of logic, doesn't it? In the sense of, if there's more competition among individuals of different species, then those individuals within a species start to specialize. But once they start to specialize, there becomes more competition within individuals of the same species. As you're trying to get after one secure resource, as an example, those that are able to get after that resource better do more effective jobs than those that are not. And therefore, an argumentation for a lot of evolutionary biology is that we are driving ourselves to extinction through specialization. That as we become more and more specialized, we become more and more sensitive to any environmental variation. And when the environment does vary, and the environment always does vary, that can lead a highly specialized species to extinction. And that's the Red Queen hypothesis. For every step forward, you take 2 steps back. Hypercompetition between different species leads to hypercompetition within individuals of the same species, which leads to high degrees of specialization, which leads to the inability to accommodate environmental variation. And in some places, this works. Hyperspecialization is a key feature of rainforest environments. One of the reasons we have so much biodiversity down there. Now, can anyone think of why specialization would work in this type of environment, like a rainforest? One, the environment is the same all year round. Can anyone think of another reason? Because that's a great observation. There's so many niches because of that hyper-specialization. So as an example, we were just down there, and I didn't bring a picture today because I didn't know I was going to talk about Van Valen. That started about 5 minutes ago. I was like, ooh, this is a good time to talk about Van Valen. But we were down there, and I am from Ireland. I took a genetic test. It's about as boring as you can get. Irish, period. And that's a lie. There's like a tiny bit of Scot. Ooh, look at all that interesting variation. Nope, that's all we're from. Fine. But you put an Irishman in the middle of the Peruvian rainforest, and what are they going to do? They're going to die, yes. Yes, thank you, David. I appreciate that. That was lovely. I like how you try to say that quietly. Alex, you gave me the clue. I appreciate that as well. What are they going to do? They're going to sweat. And I mean, just buckets, grotesque buckets. Thank God I'm married, because if I weren't, I would not be an attractive specimen, because it's just pouring. So I'd go in the field and every time come back, take off the clothes, put on a new set of clothes, put my clothes up on the line. There is a orange orchid bee that freaking loves my scent. Because as soon as I put that clothes up on the line, you have about 100 insects that I've never seen anywhere just show up on that shirt and just start consuming all of the salts coming right off of it. And I say, my sweat, because we had a bunch of shirts up on the line because we're all a bunch of gringos going down to Peru. All of us are sweating, maybe not to the same degree that I am, but there are no orchid bees on anyone else's clothing, just mine. Hyper specialization. One reason is because the environment is relatively consistent from one year to the next and one day to the next. Another reason is that environment doesn't show any evidence of changing throughout millennia. So where interglacial maximas were going up and down over the past few 100,000 years, no changes in the Amazon, none. No changes in Congo, none. No changes in Indonesia. Those environments stayed stable. They stayed rainforests, which gave that much more time for specialization. Why don't we see the same degree of specialization in environments like Florida, New Mexico, California, Canada? And its first point, environment changes constantly from day to day. You know, I'm going up to upstate New York this weekend, see my daughter. Today, -7. Thursday, high of 45. Saturday, high of -9. I mean, that is just stupid, really kind of dumb for a Floridian's perspective, back down to stupid and crazy. What's the other thing that's true about these environments? Caitlin? Have they been the same for millennia? Nah. At one point we were underwater, and another point right where we're at 10,000 years ago was 30 miles inland. As the sea levels were much lower because they got locked up with ice and the coast 30 miles that way. So massive changes. These were relatively grasslands. If you go to the Peace River right about now, it's a great time to pick up Macedon teeth, Macedon lakes, because those animals were common here. It was also cold. If you went into Canada, well, you wouldn't really go there because there was a mile of ice. So the time for things to evolve has been much shorter relative to something that's been relatively consistent. So based on Van Valen's prediction, which really does seem to hold up, competition leads to specialization. Specialization ultimately leads to extinction because of changes in the environment. If we look at these brood parasites, which one would we expect to be the most derived? Which one would probably be the ancestral condition? And for that, Miranda, which of those would be the most derived and which would be the ancestral based on Van Valen's expectations? Van Valen would actually predict this one as the ancestral because competition over time leads to specialization. So the one that's most derived would be the one that has specialized on the fewest number of hosts. In this case, Rufilixarius has specialized on one host. That's what it would predict. But that's the nice thing about theoretical predictions. We test them because Miranda's statement is exactly the way it occurs. We might expect specialization to be the most derived condition, but when we actually look at the phylogeny, what we have is the ancestral, like Miranda told us, is Rufous marius, and then what we have is increased generalization, an increased use of additional species, which then becomes an interesting feature of itself. Why aren't we seeing the Red Queen hypothesis? And that answer has not been resolved to the best of my knowledge. So we expect increased specialization, shows up all the time. But in this case, in terms of brood parasite, we actually see the exact opposite. We see evolution towards generalization so that these two species are actually using more different species of hosts. Now, can anyone think of an adaptive explanation for why they might use more different species of hosts? Are people getting on Telegram today? I know we were blanked out yesterday, but are people getting onto it today? The internet? A little bit? Okay. You are? Okay, that is good. Thank you. Can we think of why we might see the evolution in this direction? Well, you can imagine from the host perspective. Is brood parasitism a good thing or a bad thing? It's a bad thing. It's a terrible thing. You lose your chicks, one, that's not good. Second, if you are parasitized successfully, you are going to expend a tremendous amount of energy and resources to raise the offspring of another animal. So not only do you lose your offspring, but you're also wasting energy to rear someone else's offspring. So one of the things we do know about these two is the fact that they do have additional species that they can parasitize, reduces the learning of any one species to the potential for being parasitized. So that if you are being parasitized a lot, you can learn to it. And if you are being parasitized a tremendous amount, you can co-evolve responses to it. But by having so many hosts from these species, that makes sense because then they can avoid any one species picking up on them. Questions with that? That's a great question. And the answer is we don't have any evidence of that occurring yet. What we do have evidence of is the brood parasites are more successful with naive parents, first-time parents. And they are less successful with older parents, parents that have already had offspring. And there is some evidence in that case of learning, but as you're kind of suggesting, the system is still operating, even though Rufous marius is only attacking one species. And that's not entirely clear. Some hosts can. Some hosts can recognize the number of eggs. Some hosts can recognize the fact that the egg looks fundamentally different. And some of those images we saw from the videos, the eggs were white, where the brood parasite's eggs was very molten. And they can pick up on those signals over time. It's a cool system. Other questions? Okay. So what I want us to do is I want us to play around with this phylogeny. And what I want us to play around with is grooming behavior, with our different types of ways in which these insects can groom. And this is another video that someone shows some science in terms of so what they did is they attached a little barrier, almost like a dog collar, around the antenna to keep the animals from grooming one or two of their antennas. And then they looked at how they were able to identify their chemo and mechanical environments relative to grooming. And that is cockroach, and that is Blattaria. What we're looking at is grooming of mouthparts. So in terms of these character states up top, you have the different traits: grooming antenna with mouths, grooming forelegs with mouths, grooming hind legs with mouth. For each one of these different groups, Coleoptera, these are beetles; Diptera, these are flies; Blattaria, these are cockroaches; and Orthoptera, these are grasshoppers and katydids. They have either a plus because that's what they do, like we just saw that American cockroach, Blattaria, grabbing its antenna and grooming it, or a minus, where in this case, Diptera, they do not grab their antenna and groove it with their mouth parts. So in some cases, we have pluses across, in other cases, we have pluses and minuses depending on this arrangement. This is an appropriate phylogeny for these 4 different species. You have the outgroup, then you have Diptera, Coleoptera, Orthoptera, Blattaria. And what I want you guys to do is I want you to use both combinations of the character states as well as the phylogeny, describe a little bit of the evolutionary history of these traits, where they emerged, what they're shared by, sort of a reinforcement of homology, homoplasia, convergence, and evolutionary steps. Make sense? Very good start. You have a table by yourself, I'd encourage you to move.
Welcome everybody.
Okay. So I know most people's opinions of insects, especially most people's opinions of cockroaches, are not what we're going to call good. Most people do not like them. One of the things to recognize about these animals, though, is they are psychotically clean. They spend a very significant amount of their lifetime cleaning themselves. I expect one of the reasons that we're not big fans of them, especially things like the American cockroach and the German cockroach, are the two most often we see in our environments, is not because they end up being dirty, but they kind of reflect ourselves as maybe having dirty conditions, because cockroaches are great at cleaning up our mess. When they come in, they're just eating up the food, maybe it's like a little sugar residue where they want to counter, and they're just going for it. They love it. Absolutely love it. So, second thing before we get to these, I'm going to try to find these abstract videos. These started maybe 10 years ago as a lark. And one of the first ones I found was a paper that described a preference for fermented nectar in honeybees, which just process that for half a second. First, I'm smart enough to do that study. That's kick-ass. A study about preference of honeybees for fermented nectar. Two, after doing the study and finding that these honeybees do have a preference for fermented nectar, and just like others who take fermented liquids, it causes disorientation in honeybees, but they still go for it. To actually video it, bring it out as a video abstract. This is happening a lot more, and with the advent of the pandemic, I'm now seeing it a lot more. How many of you are involved in some sort of research project or another? How many of you expect to be presenting sometime in the relative future? How many expect to do it in a virtual environment? Where we're going to be sort of presenting not in front of people virtually, a few. If you are, let me encourage you to do a video abstract instead of the traditional virtual talking in front of a PowerPoint. First, how many people like PowerPoints? They're like, ooh, hey, can I see another? And I say that, I use them heavily, and I do well with them heavily. They keep me off my shiny object syndrome, like that's an example. When we spend the first half talking about Van Valen, not because it was there, just because that's where my mind went. If I don't have that, my mind will just go all over. And that sort of tempers my direction. But most people don't really like to just sort of see a slideshow. But a video abstract is a really powerful way to communicate what you do. You can visually show the methodologies as you're doing it. You can visually show the system. In addition to your results, you'll have some slides that are really PowerPoint slides with some graphs and stuff like that. But then you can also visually show the context both for why you're doing it, the intro, as well as ramifications of it. And it provides a clean, good visual narrative. It's a really powerful tool. And to give you sort of an example, how many people don't trust science systems? Like, literally. We're like, yeah, science knows what it's doing. It's cool. Any evidence that maybe that's not universal? Maybe in this country? Are people like science? Yeah, I don't know. Really? Nothing. It is not Monday, it's Tuesday. Good?
The whole pandemic vaccine.
Whole pandemic vaccine where people are like, maybe. My Super Bowl Sunday, I somehow got the end of the table as we had 12 people in our house, all the grandparents, and I got the crazy side. We went first vaccines, and then we went ghosts. It was just awesome. And an entire night there, I just sat and just ate my barbecue, which was delicious. I enjoyed my barbecue, but I said nothing, as they were just off. What? Any other evidence where science is like, I don't know. Climate change, I don't know. Anyone see the report that came out this morning? I don't know this morning, so you've had a lot of time to catch up on it. But there was evidence that got reported yesterday that the Southwest United States is in the worst drought since 800 AD. So for the past 1,200 years, Southwest United States is in the worst drought ever recorded. And they use dendrochronology, which is where you do tree port samples, and you can look at the width between the rings in a tree to determine how much growth there is, with a primary factor influencing that growth being water. And when you have periods of drought, you get very little tree ring growth because there is no growth. It's sort of basically stagnant. And that this right now, the drought they're experiencing in Nevada, Arizona, New Mexico, California, this is the worst drought that has ever been reported since the time of Charlemagne. And Charlemagne was the king of the Franks that led to the beginning of the French nation after the downfall of the Western Roman Empire. So that's a long time right now. Now, maybe that's climate change. Maybe that's just random climate. And a variety of things, not totally clear about that particular episode. But crazy stuff. Yeah, so you're like, maybe not. This, you get good information from it. Use the videos if you get to make it. And that's the point of sort of the science thing. Talked to a guy last weekend. He took one of his scientific papers, made it to a graphic novel. Process that for a second. About evapotranspiration in urban forests. Worked up with an artist to make it into a graphic novel. Do you know how many people normally read a scientific paper? Tens. Like literally tens. They take so much time to write, and maybe tens. My wife says she read one of my papers. I don't believe her. I don't think she ever has. And she's the only person that may have read anything I've ever written in my household. That's how many people read this graphic novel. 500,000 hits. It was streamed up. It was read. That's insane. So if you can find other ways to share out your science besides just your traditional circles, you do the traditional circles. But these other avenues can be much more impactful because more people are willing to engage. Oh, yeah, okay, done. See, and that's not in the PowerPoint that I got off again. 2 times today. So we'll try to stay on. We'll start with Corey. Corey, you got the first softball. What species most related to Coleoptera and why? Yeah, Diptera. Diptera. And why? They branch off together. They share the most recent common ancestor. Perfect. Love it. And what's Diptera? Diptera? I say Diptera. You can say Diptera. It's tomato. What's Diptera? No! Diptera is the cockroach. Anyone know Diptera? They're flies, which include mosquitoes and crane flies and midges and all that. Coleoptera, anyone remember Coleoptera? It's the beetles. So anytime you think about Ringo, you can think about Coleoptera. And then Orthoptera. What is Orthoptera? Grasshoppers. Is that going to be on any test you get? No, got it. No, that's not. But I still will repeat it. Okay, so then we go to Shannon. You're up next. And which of these characters is pretty uninformative? Not informative. Doesn't tell us anything about relationships, and it doesn't tell us anything about the evolution of the trait. Perfect. Grimm's 4 legs and mouth because he's universal across. It doesn't inform us about the tree, and we don't get any information about that particular behavioral trait within this biology. Perfect. So then we come down. Lewis, welcome. How you doing, buddy? Is absence of grooming antennas with mouth parts homologous or convergent between Diptera and Orthoptera? And why?
I said homologous share between phylogenetics, even though they're branched off separately.
Okay. So where do we see grooming of, where is it? Grooming antennas with mouth parts. We got a right here. Coleoptera has it, right? And Orthoptera has it. Is that right? Yep, right. Okay. What does the outgroup tend to be? What does the outgroup tend to be? Why do we add outgroups? Do you remember? It's like a control. So if we see derived characters emerge and the outgroup tends not to have it, so we start out with not to have it. Okay, so now we have a few options. We can either see evolve and then get lost twice. That's 3 steps, right? Or we can see it not be present, and therefore that's why we have it not here, and therefore evolve twice. Which one's more parsimonious, 3 times or 2 times? Parsimonious. 2 times. So, which is the most likely explanation? Most likely explanation is that they are convergent and homoplasy because our outgroup does not have it, and therefore it's likely that they both evolved independently. The alternative could be true, it's just not parsimonious. That is, our outgroup does not have it, it evolves once, and then it is lost twice. So they're both reasonable hypotheses, but generally we drive the assumption of parsimony. And then last but not least, Caitlin, name the character state that is derived from homologous between Diptera and Coleoptera. Grooming forelegs with mouth is everything. Yep, and they do not have it. Yep. Good. Any questions with that? Okay. Oh, listen. Did you say you schedule the breaks? I try to take one at 45 minutes, and then we're roughly the hour and a half mark. So come on back at 8:20. Okay, so the evolution of some traits is just fundamentally weird. And the only way to sort of get at how something like this can occur is through a phylogeny. Anyone enjoy themselves yesterday? Whether it be through a romantic day with your loved one, whether it be through a galentine, or whether it be through a bromance, or whatever it be through whatever you need. Did anyone enjoy themselves yesterday? Nope. Oh, what's that? My wife did not, but I have a beautiful wife, and I did enjoy myself yesterday because she is good. I am terrible and negligent. We've established this a long time ago, and I reinforced that where she got me a gift to go see. And I don't know how this arrangement's going to be working, but it's a orchestra of David Bowie and Philip Glass's Concerto Number 4, and I'm sure everyone knows David Bowie, right? Does anyone know Philip Glass? Freaking awesome. Check out Metamorphosis, one of the most beautiful, haunting pieces of music, a minimalist pianist, mostly through the 60s to now, still one of the most important contemporary composers. And it's going to be a weird show, but she'll hate it, which reinforces how much she likes me. So, yay. But that's the whole point. That's a really stupid thing for her to do. Put herself into a terrible situation because she hates modern classical music. And if you ever really need to know what modern classical music is, Come in sometime. I'll play you a little NC. You can choose between Baby or Molly, and they're both phenomenal performances. Baby is about 5.5 minutes. Molly is about 42 minutes. They're incredible. They're the exact same piece of music, but it's this improvisational orchestration within C major, and it is astonishing if you like it. Unfortunately, most people do not. So most people, it is this mind-crushing fingernails on a chalkboard type of experience. But if you want to know what modern classical music is, just come on, we'll just enjoy it together for a little while. If you walk out of the room in 30 seconds, I won't be totally surprised. But still, you can have some fun with it. Because some of these things are really, really dumb. And these are things like nuptial gifts. And a nuptial gift is something associated with courtship, much like Valentine's Day, where a gift is provided that is supposed to reflect some value in terms of the courtship. And some of these nuptial gifts can have value. You know, if commercials are any indication, we're supposed to buy our loved ones a car on Valentine's Day. Well, a car has value. It can be for a form of transportation, which is of value. Or you could take that car and say, thanks, I already have one, and sell it, which is of value. And both of them provide very clear value. Some nuptial gifts do not. And we engage in this very weird nuptial gifts as humans, where in the process of courtship, if we want to finalize courtship, when we stop experimenting and looking around, typically the male, not always the male, will go out and use a massive amount of resources to buy a rock and put it on a ring, you know, a little loop, and then provide it as a gift. And it's normally a gift of an acceptance or a rejection. And this like just nugget on a circle has no value whatsoever. We prescribe value to it, but it has no value whatsoever. And it is a nuptial gift where the overall maybe quality of the rock and the loop, or the amount of time for the rock and the loop, is supposed to reflect something about the gift. Does that make sense? So like my rock and loop was not terribly expensive in terms of finance, but it took over 6 months to put together. Is that we had to find a pearl from a particular sea, which took a few months to go find that. And then we had to go send it out to get engraved based on all the things that she talked about our first date, because she did not shut up the entire night. She just talked incessantly, because I took her to a place that was terrifying called the Amalga Barrens. I didn't really think this through very well. And she at that point decided I was the axe murderer she thought I was going to be. And that maybe if she kept talking, I wouldn't actually bring out the axe. But I did bring out a big, huge Bowie knife, because we had apples, and that didn't process until I actually had the knife in my hand that I pulled out of the pickup truck that was leaning at a 45-degree angle, that this might terrify her. So I gave her the knife, and again, she just kept talking and talking about all these random things. So the ring, this thing that has no value, is sort of inscribed with all the sort of topics that she engaged in that night. Okay. No value in it though, because can she do anything with it? No. Can she care for her offsprings with it? No. Can she purchase a house with it? No. Can she get food for it? No. She's tried to eat it a few times, you know, thinking it was like one of those little, what do we call those, lollipop rings, which do have value. You know, you can put that one on there, and then you get at least a good 30 minutes like, and that's value. But this one, no. And then we provide it. We provide it. Now, that is a ridiculous gift that we provide, right? We all agree on this? Yeah, yeah, true. Good. Thank you for the head nods, whether you agree with it or not. I do appreciate it. Scorpion flies have us beat. I love scorpion flies. First, the scorpion tail is their genitalia. It's not a stinger, which is kind of weird. Two, at least one species of scorpion fly provides a nuptial gift that is a balloon. Now try to process that. I'm going to agree to mate, and you're going to give me a green balloon. That is a hollow empty nuptial gift. So let's first start by taking a little bit of a look at these guys, because they really are cool. And a lot of them also nice things.
Scorpion flies. Funny how most of the good clickbaiting names come from insects. The name scorpion fly refers to an entire order of insects, Mecoptera, with over 600 individual species and 9 families. So technically they aren't actually flies, they are their own order of fly-like animals. The scorpion part comes from the obvious scorpion-like tail with its large stinger-looking thing. It's actually not a stinger, but the class the male uses to mate with. Essentially its genitalia. Being an entire order of animals means that they live almost everywhere, minus Madagascar and a few small islands around the world. They're found in Japan, Taiwan, and the UK, but it's believed they are able to reach these areas due to land bridges that once existed to these islands. They are also present in Australia, however this continent has not had a land bridge to it in the recent 100,000 years. But Mecoptera is very old, dating back to the Upper Permian, and so it may have just been on Australia the whole time. Some live in desert conditions, and even some in cold, snowy climates. But most species of scorpion fly inhabit moist wetlands, grasslands, and woodlands, and thrive in the wet leaf litter. The leaf litter provides an excellent environment for these insects to feed in. Scorpion flies have a very wide-ranging diet due to how widespread the order is. Some scorpion flies are carnivorous scavengers, feeding upon dead insects amongst their leaf litter, and others eat flies stuck in spider webs, and sometimes even the spiders themselves. Hanging flies, a family of scorpion flies, feed by dangling their long legs and catching insects mid-flight. Other scorpion flies prefer to feast on carrion and others' larvae. As well as scavenging all this meat, they may also consume decaying vegetation, nectar, and even pollen from flowers. What they do and don't eat depends heavily on where they are in the world, with desert-dwelling scorpion flies obviously not being able to feed on moist, decaying vegetation. The mating process with these animals is actually rather nice and far less aggressive than many others. Males will secrete pheromones to attract females, and then most males will produce some sort of edible gift for the female, such as a caterpillar or a ground secretion that the females will consume if it wishes to mate. The larger the gift of food, the longer the male has to mate, and the more successful it will be. As soon as the female starts to eat, the males will use its stinger like appendage, which is actually its genitalia, and grasp onto the females behind and begin to mate while the female eats her snack. The female will lay eggs in moist areas as the eggs need to absorb water and expand. Of course, for those living in arid conditions, the eggs will hatch after the dry season is finished, which can mean they stay as eggs for many months. The larvae will look like weird caterpillar things, and just like most insects do, they spend the early stages of their life foraging on decaying vegetation and insects. Interestingly, when they enter the pupa stage, they do not go into a cocoon. They simply bury themselves in soil or decaying vegetation. Once again, in arid conditions, the same is carried out, but over a longer time frame. Well, the obvious one, the scorpion-like stinger, has already been explained. It's simply there to house the male's genitalia. The females do also have tails, but they do not possess a large stinger-like thing, only a hole where the male puts their stinger-like thing. An interesting piece of behavior, which isn't exactly an adaptation, but is too interesting not to mention, is their habit of eating human corpses. As they're scavengers, they will eat really any meat they can find, which includes human. Don't worry, they won't attack a live human, but a dead one is an easy meal to them. This tendency to eat human flesh is incredibly useful in the field of forensics, as it was discovered that scorpion flies are the first insects to arrive to corpse and will stay there for a day and a half. This allows forensic teams to date bodies in crime scenes very easily, which can help massively in criminal investigations. So really, we should be thanking these little flies for their service to justice. Just ignore the fact that they eat dead humans. Scorpion flies are predatory, and so other insects tend to stay away from them. But being so widespread around the world means that they must run into some sort of trouble. The stinger-like appendage is likely to ward off some predators, and it certainly keeps humans away, despite the fact that it's completely harmless unless you're already dead. The extent of the order means it's unlikely to go extinct. It lasts since the Permian, so it will probably last longer. Also, it's an entire order, and it's really hard for an entire one of those to go extinct, but individual species certainly can. Thank you for watching this video. I really hope you.
So he ended up talking about the nuptial gift in terms of, let's call it a logical, clear reward. We do have a lot of species where the male will capture a prey item, and the male will take that prey item to a female and offer it. And kind of like the redback spider from before, if she accepts it, then the male has a longer opportunity to mate while she consumes the food. And this nuptial gift actually makes a lot of logic in the sense that not only is it a gift, but it actually provides a direct value to her mate, his mate. And she is going to have to produce all the eggs, to which lipids and proteins are limited resources that are necessary to produce that effort. So this makes perfect sense. How, though, do you get a situation like this, where literally, instead of the male finding a prey item, the male builds a cocoon. Remember, they don't build a cocoon when they pupate. So that is not part of their life history. But in this case, the male builds a cocoon. The male provides this balloon, which is just this hollow out cocoon, to the female. She evaluates the balloon, which has nothing inside of it. And if she likes the balloon, they mate. And if she doesn't like the balloon, no. How does that occur? And how cool is that? And if you remember, one of the things we talked about at the beginning of the semester, and as I keep mentioning, animals will do things that really don't fit our expectation. It doesn't mean it's not real nor unreasonable. Redback spiders are weird as haggies. They're crazy. That facilitated cannibalism. But it occurs, and it makes sense. Females accepting a balloon as courtship to sire offspring? Yeah, that's weird. But it works. So how? This is where phylogeny can become really helpful. So instead of looking at a tree, what we're actually going to look at is we're going to look at the steps involved in the process. What we have is this derived species. The derived species is the one that presents the empty balloon. How does this come to be? Well, what the video talked about is sort of the first step and the most widely observed case. Well, actually, no, I'm sorry, not the first step. The ancestral condition, there are a few species, not many, but a few species where there is no nuptial gift involved. There is just traditional forms of courtship. Maybe it's associated with a dance, maybe it's associated with pheromones, but just traditional form of courtship. The male approaches the female evaluates the male. If she either rejects or accepts, if she accepts, they mate. Nuptial gifts came in, and this is the most common form where a male captures a prey item. Now, again, they talked about a lot of the species being carrion eaters, but a lot of species will also consume live prey. The male captures that prey item, provides it directly to the female. The female evaluates the size, as an indicator of nutritional status, looks at the prey. If it's acceptable, they mate. If it's not, she rejects them. That makes sense, right? Good gift for the female, good presentation from the male. Now, in a lot of environments, and this is kind of alluded to in the video, these are difficult environments and things may decay quickly, like in a rainforest. So in some cases, the male captures a prey item and then wraps it in a cocoon, essentially a preservative, and therefore can keep that prey in a state which would be acceptable to a female for a longer period of time as he goes searching around with this basically wrapped up prey item to present to the female, and the female evaluates the balloon with the prey item inside to decide whether to mate or not. And this is when the males have the opportunity to parasitize. In at least one species, the female developed a preference not for the prey item, but an associative preference for the balloon. So instead of evaluating the nuptial gift as an indicator of what is inside the prey item, the female evaluated the nuptial gift as what was observable, the balloon. And that preference became evolved. Once that preference became evolved, all you need is one male not to present a prey item inside the balloon. And it doesn't matter anymore because that male doesn't need to give a prey item. The female is going to basically identify the preference based on the structure of that balloon. The structure is of sufficient size, then she mates. And thus, the males are now parasitizing with a balloon. It's kind of cool. Questions for that? Another situation we talked about are traits where we can't test these with experiments and or observational studies. And you can't run an experiment or an observational study when there's no variation. And a good example of where there's no variation is the nesting behavior of cliff nesting birds. There's no variation in the overall behaviors. They all basically do the exact same thing. And as we'll see, makes a lot of sense. So if we look at something like the black-legged kittiwake, I'll show us 2 or 3 times. What I want you to do is pick up on some of the behaviors of the adults. I really want you to pick up some of the behaviors of the young and some of the interactions between the adults and the young. These are a northern seashore bird. They are cliff nesters. And focus on some of the behaviors that are there and some of the behaviors that are not there. I'll show you this one more time before showing you a ground nesting bird. And while you're watching this, I'll make a little plug for open source information. This was an open source effort called Archive, where they tried to collect natural history images and natural history videos of all variety of different animals. It was open source in the sense of you could freely upload, freely make use of, especially for educational purposes. So it's kind of a lot like Wikipedia. Unfortunately, these guys didn't get enough donations, enough funding, and shut down about 3 or 4 years ago. So for things like open source, if you do feel a like of it, you do feel a compunction, do 2 things. 1, contribute information. And then 2, if you have the means, provide a donation. I think Wikipedia is only a staff of 5 to 7 people, period. It's all volunteer contributions. And it's probably one of the most powerful things we have available to us. This one didn't survive. And you guys know what open source means? Can someone tell us? Kai?
Are like basically the ones who kind of upload and manipulate all data in private.
Yep.
For example, like our video is, or our, yeah, our video is a great one, or like coded content.
Yep. So it's any type of technology that is freely available and produced through voluntary efforts. R is a statistical language that we won't use in this class unless you want to, but it's open source, which means it's freely available, accessible to everything. We're doing more and more open publications where the publication is freely available. It's not hidden behind a paywall. If you use Open Office, it's a free open access form of Word, Excel, very strong, very powerful. These are great, but they're all voluntary efforts. So if they make it because of volunteers. If they don't, because there weren't enough people to contribute. Archive didn't have it, unfortunately. Now these are raw skulls. I want you to pay attention to the kittiwakes, which are cliff nesting, versus the raw skulls, which are ground nesting. Again, pay attention to the behaviors that are there, those that are not there. Interaction between the parents and the young. Pay particular attention to the young. We'll do that one more time. So Kyle, let's start with you. Anything you notice?
Not like they were combining, but they're like sharing. Like the photos were data.
Okay, so in the kittiwakes, the food is being directly transferred into the young's mouth, and the young actually gets its beaks up inside the parent's mouth. And the second one, the raw skull, the adult just regurgitates out the food, and then the young peck at it. That's perfect, love it. Nicole, what'd you notice? Very dense, very, very dense. Now, sometimes you get that in ground nesting birds, but that is true, and especially in this one, it's more spread out. It's a good observation. One of the things with cliffs is cliffs tend to be a limited resource. You know, there's more open flat ground than there are cliffs outside of like cities and things. But great observation. What's another thing we noticed? Virginia. What about the nest? You notice anything about the nest? I'm so sorry. There's twigs and moss. Let me see if I can find an image of it. Whoops. Okay, any difference you notice between the nests there? Not a perfect image, so I apologize. So this is more grass. What else do we notice about the nests? And they're both eating grass.
Like more compact.
More compact, it's denser, and it's deep. So in the kittiwakes, the nest is very deep. You know, there's high walls. In the raw skull, the nest is more cup-shaped, more flat, kind of a saucer type shape. Anything else we notice? Anything with the chicks? And Audrey, I want to give you first chance at this. Any difference in the chick behavior? I'm going to give you a little bit of raw skull because I think we get some good here. Yep, that's pretty good. And that's pretty good. Any difference you notice between them there? Absolutely. The kittiwakes are very stationary. They don't do wing movements. They basically stay there, where the raw skulls, even in that feeding, are kind of jumping around. You see their wings going up and down. Great observation. And maybe one more thing. What directions do the kittiwake calls face? David? Perfect. They face the wall. Where the gulls, you didn't get a lot of video to see this, so it would be pretty easy to miss, but they'll face in any direction. And there's a variety of things that sort of look like they're fundamentally different between these animals. You know, the ground nesting gulls, it's kind of a shallow cup type structure. You guys noted very nicely, the young are fed by regurgitation, the adult just regurgitates out the food, and the young peck at it. One of the things you wouldn't be able to see or notice, but it is definitely true, is a strong parental recognition. The gull chicks know who their parents are. Their claws, their feet are relatively weak. You know, and you kind of saw them bouncing around, but they don't really have strong claws. They do engage in a lot of wing movements as they have the downy feathers. They're kind of building up their breast muscles and getting prepared for eventual flight. And they face in any direction. They just sort of circle around no matter what. By comparison, you guys are beautifully picking up on this. One, the nest is a very deep cup. It is a long cup, and the note, and I think goes good too. It's more dense. You know, it's the same type of material, grasses, but they're weaved in a much more dense way. The young are fed directly by the throat. The young gets its mouth up into the parent's throat to get it. This one's a little surprising. We don't have evidence of parental recognition. They just behave like that to whoever shows up at their nest. The parents know the chicks, but the chicks don't know the parents. And if you've ever been a parent, you'll get this once or twice in your life where the kid's like, yeah, I don't know, they're kind of cool, but that one's probably more my parents. You'll just walk away with that. The young have very strong claws, which kind of makes sense for being. And they have very weak flight movement and flight muscles. They don't engage in that. In fact, their first flight, I'm a little terrified. They just get out and they fall. And if they're able to get lift and they stay. And the qualifying statement is if. There's a lot of chicks that go through this first jump. Another thing we see, this is universal. Now, we can reason out that that is an adaptation for a cliff, right? We can reason out that's an adaptation for cliff nesting. We can reason out these are adaptations for cliff nesting. We can reason out this sounds like an adaptation for cliff nesting. Can you do the experiment? Especially that last one. Now, the first way you can do an experiment How can you do an experiment where you look at the cup shape to determine whether it's adaptive? Anyone think of a good experiment? I didn't say effort. I just said good. Those are two different things. What would be a way in which we can experiment? Good.
Where some are equal to R and see which one they use.
Perfect. You can set up multiple nests, see where some are deep, some are not. Look at which ones actually hold eggs. So not which ones they use because they make their own nest, so they won't take something that you make. But you can see which ones hold eggs. That'd be a fun experiment, which is totally ethical. But then you can also get real nests. You can add to the nest of some of them, keep some of them as control, and take away from the nest of some active nests. That's the unethical one, where then you can test how those eggs survive, and presumably some of the eggs on the more shallow nests would not. But it works. It's a good experiment. Can you do this experiment? Feed directly by throat. You might be more creative than me, but I can't think of a way to do it. It's universal, and I can't think of a way in which I could manipulate the parents' interaction with the chick to do it. So it's kind of beyond our control to study, unless you come up with something to tell me. That'd be cool. Parental recognition, I can't see how we could do an experiment there, because if they don't show it, there's no variation. So it's not like you can get some to show it, some not show it, and look at their overall response. The weak claws, you might be able to sort of take a claw, and again, maybe not ethically, but damage it or change its ability to function, making it weaker, and look at the response of those chicks. So you might be able to do that one. Weak flight movement, that one, because I don't know how to increase the flight movement. Maybe you could put like a little electrical probes that sort of force the chick's arm through sort of stimulation to do it. Again, not ethical, but may work. What about this last one? Young facing the wall. Can you think of any way to do an experiment here? I think I can, or I can't think of like how I can force the chick to face the other way. Did it? That's interesting. Maybe put them in a mirror, maybe see if they face the other way. But you probably need to put something on the other side to show them that that's a cliff. So if they face that way, then that doesn't tell you whether, it tells you that they want to face a cliff, but it doesn't tell you why they're facing the cliff. But I like that idea. Are they actually noting the cliff? And you can look at like, what are the signals they're picking up in the cliff? It's very clever. Okay, so we have a problem. For at least this last part, we can maybe look at the mechanism behind it, but we can't quite figure out the adaptation. This is where comparative methods can be very powerful. And what you can use is a phylogeny of distantly related species, specifically looking for homoplays. Where if you have two species that are distantly related, that are convergent on the same behavior, but do not share a common ancestor relative to that behavior, you have pretty strong evidence that the behavior is being done for the same reason. And a good example of this are kittiwakes and our swallow-tailed gulls from the Galapagos. Both of them are cliff nesting. Both of them feed directly into the mouth of the chick. Both of them, the chicks don't do strong wing movements. In addition, the chicks face the wall of the cliff. So if they do share a common ancestor, that's homology, and we don't know why it occurs because we don't know the condition of the common ancestor. But if they are not sharing a common ancestor and they show both the same traits, then we have pretty strong evidence of adaptive convergence. And to illustrate those two different hypotheses, in this case, we would have pretty good evidence that the cliff facing, the weak wing movement, all the data associated with being a cliff nesting bird is adaptive. They don't share a common ancestor with that trait, and therefore it's likely they both converge on the same adaptation. But in this case, if it's a tree on our right, we would not have direct support for the adaptive function, because again, we wouldn't know what the condition was for the ancestral species. That makes sense. Anyone know which of these two trees is more likely, and therefore whether this is convergence, and therefore adaptive or not? In this case, it's a tree of life. Those two birds do not share a direct common ancestor that showed cliff nesting, and therefore it does look like, in this case, facing the cliff wall, deep nests is all divergence and adapted for nesting in these cliff environments. Questions with this? Okay, so another example of trying to figure out something that you really just can't do with other methodologies. You know, why are some species of ungulates spotted when they're young? I think I'll show you this later in the semester. Pronghorn antelope, when they're young, they're not spotted at all. But when you look at white-tailed deer, they're young, they have little spots on their back. And if you know white-tailed deer, as they mature, those spots disappear until it's a relatively uniform back color for their fur. The belly is slightly different than the back, their tails are very different. And you can also ask the question, why are some ungulates striped? With probably the most famous example being the zebra. The zebra has a very distinct black and white pattern. And we use phylogeny to sort of test these different hypotheses. So this is a phylogeny of ungulates, and do not worry, you cannot read that. You cannot read that in the paper, even when you have good resolution, the magnifying glass to look at it, and you honestly don't care. That's not what the tree is for in this case. What the tree is for in this case is to test a hypothesis about the adaptive function for the spots. And what you're looking at is two different cases. We're looking at the color of the line in the phylogeny, whether it's an open white line like you see right here, or whether it's a black line that you see right here. And again, that is a relatively difficult thing to see. If it is a white line, then what we have are young without spots. And if it is a dark line, then what we have are young with spots. And what we're doing is we're comparing that to the behavior of the animal. Do the young hide, or do the young move actively? And how many people have seen a doe and a white-tailed deer young interact in a forest? Anyone have a chance? Can you describe some of the things you saw?
Oh, they were everywhere in New Jersey. But, a lot of them were surprisingly like desensitized. Like if you were, like I would usually go ride my bike down like by the woods, and I'd stop and like watch like some deer, and they looked right at me, but just continue doing what they were doing. That was really cool. Actually, one time I had a baby deer that was hit by a car, walk up to my front door and wait there, and they would do that for like 3 mornings when I was waiting for the bus until I finally took it to like the rehab center. It was very interesting because they don't see, like, of course when they're in front of the car, that's fine, but when they see humans, especially in a place like Jersey where they're so common, they're really not scared at all.
Yep. And one of the things, and this is actually true pretty universally, you can tell a lot about a local community based on how the animals respond in that local community. On this campus, when you walk around, the brown and knolls, which we will work on this Thursday, they don't care. So long as you behave like a normal human. So long as you stay on a pathway, so long as you sit on the spots associated with humans, they don't care. If you change the typical human behavior, like instead of walking on a pathway, you walk directly at the knoll, or you change the typical human behavior, instead of sitting at a picnic bench, you sit right next to the tree within a knoll, then they care. But they don't care otherwise. Are squirrels on this campus? They don't care. More often than not, even when they're barking, they're not barking at us. They're just sort of barking at something else. They're good about a call if a hawk comes by. But if you come by, they don't care. Unless you stop and you look directly at them, and then they care. So as long as you behave like a normal human behaves, fine. And to use the example of the squirrels, if you go out to Alba, you go out to LaBelle, you go out to, what is that little town directly south of Lake Okeechobee? Lewiston. Going in those places, the squirrels are not going to behave the same. They do care. You come near the squirrel will move. You approach the squirrel will go into a tree. They don't care. Those animals can tell you a lot about how humans interact with them and whether we are perceived as a threat or not. On our campus, so long as you behave like the traditional human, nothing. If you behave like a non-traditional human, then something. And if you go into an area where maybe there's more hunting and more direct interactions with wildlife, then you'll get differences in behaviors. One of the reasons the Galapagos is such a beautiful place to see wildlife is because they don't care. And the reason they don't care is humans have very specific locations where they can go and where they cannot go. So when you go into the forest in the Galapagos, you go in a very specific path and you cannot step off that path. And the birds are right there and the sea lions are right there. They do not care because you are not going to step off that path. If you are in the city, the lines between the national park and the city are a perfectly straight line. And humans do not go on the other side because you are not allowed to go on the other side. Period. Everything else is off limits. And then we'll just good observation. What else has anyone seen with a doe and a young in terms of like white-tailed deer? Well, one of the things will be cool is a doe and a young will interact. Maybe the young will feed, and then afterwards, the doe have the young just sit down in a spot, and then she'll leave. And the young just stays there and they hide. Lack of a better phrase. This is relatively common behavior in a lot of young ungulates. In other young ungulates, like wildebeest, the young may lay down, but the young always stays in very close proximity to the mother. So when the mother moves away, the young stays with it. So where the lines are describing whether we have spots here, no spots there, these little boxes, and again, you can't really see them, are describing whether those deer, those ungulates are showing hiding behavior or not. If the black box is showing hiding behavior, if it's white box or not. What is of importance to you is that. What is a p-value mean? Probability of what? And there's the only person who's not letting answer is Kyle. Sorry, Kyle.
It's not random.
That's a perfect technical way of describing it. It describes the probability that the pattern you see is not random. Now, that is a perfect technical way of describing it. It's also a very confusing way. Kai, what is a more simple way to describe it? So that's also a very good technical way of describing it. There's a 5% probability that the null hypothesis is actually correct.
Oh, yeah, okay. Wait.
What's a simple one?
Yeah. There's 5% chance
Simple, even more simple. I just took biostats with me. I don't think anyone else is taking biostats.
yeah.
Simple, make it simple.
I would say, I don't know.
Translate. Probability mistake. Now translate it out. Make it simple. P-values suck. Like, I love stats. I love stats. That's amazing. We'll be doing a bunch of it. Don't worry about it. Victoria, what am I doing here? What a jackass. Not you, me. What a jackass. Translate, because high statement, heterogeneous statement, variance statement. Perfect. Translate, make it simple. If it's not the null hypothesis, what is it? And what's the alternative hypothesis be? I know this is why stats suck. It's much easier than this. When the p-value is small, it means there's a biological pattern. When the p-value is large, it means there's a random pattern. Just that's a simple one. Everything I was said was very good technical information for stats. In this class, we are going to function only on the biology. We are going to simplify these things out to function in biology. We want to take biostats. It's a rocking class. I would encourage you to do it. I love it. And we'll get into a little bit more of the weeds, then we'll get back into the circle. When it's a small p-value, you have a biological pattern.
So this is saying that hiding behavior is associated with the spots. So it's making an argument that the spots is a way to, or it is a morphology associated with hiding, likely making the animal more precise. The idea that tells you about the null hypothesis, tells you about random patterns, all very true. For us, we're going to translate out in this class. Small p value means what? Biological pattern. Large p value means what? Random pattern. We're just going to treat it as those two. Small p value, biological pattern, large p value, random pattern. Both are cool patterns. But that's how we're going to translate. Make sense? Good? Yep. Okay. So this is another one. One of the arguments, it's a really cool argument, is that the stripes, patterns, like tigers, even though this isn't tigers, it's all arguments, like zebras, is associated with light forests. And what a light forest is, are forests where you have the horizontal structure of the trees broken up by light, so you get bright, dark patterns. And you can visualize it. You know, where the tree is, dark, where the light streams through, bright, tree, dark, light, bright. They get these sort of interval patterns of light and dark. And the argument is that in a light forest, you might expect to see more animals with stripes because the stripes could be reflecting that light-dark pattern, and therefore they get crypsis. Hi, do you have a question? Okay. So in this one, what we're looking at for the lines, if we have the black lines, we have striped coats, like zebras. If we have the white lines, we have non-striped coats. And if we have the black boxes, we have light forests, like here. If we have the white boxes, then we have non-white forests. Okay?
So this value, like what's a good confidence interval for like studies like the phylogenetics studies like that?
Oh.
Are they super specific or?
This was definitely for you and Victoria. This is definitely Popperian. So it's old school mathematics behind it. It's still pretty recent, but we would probably do a different procedure with it now to reevaluate. There was no use of modeling out this pattern and bootstrapping it. So it's kind of a firm, just associative pattern. And it's almost, and we didn't do this in our class, but I think all of you guys have done this in genetics, a contingency table, a g-test, a chi-square table. Yeah, that's basically what we're looking at here. Are the probabilities of the black lines associated with the black boxes more than would be expected randomly? So it's not very rigorous stats here. But in this case, do we have a random pattern or do we have a biological pattern? Would that be value right there? This one actually is better because I know you all took stats now, and I know you memorized every part of that class because I know that is the favorite class for every single student at FGCU, especially biology majors, correct? Exactly, and I can see by the smiles on your face that you are feeling all those good emotions you had back in stats methods, correct? Fantastic. Random or biological? Is that a large p-value or small p-value? That is a large p-value. So that is a random pattern. Someone remind me what the threshold is, what's the typical threshold associated with whether it's large or small? 0.05, right? So typical threshold. If the logic behind it is 2 standard deviations away from a traditional normal distribution mean. That's the logic. Cool. So in one case, we have evidence that supports the morphology and behavior. In the other case, we have evidence that does not support the morphology and behavior. In both cases, we're able to test something that would not be testable otherwise. Make sense? Cool. So two small last things. Why do we need large data sets? Why do we need to have traits like base pair sequences and or morphology? And the reason is we are trying to discriminate between an unresolved and a resolved tree. When you look at a phylogeny, if you have a straight flat line describing the relationship between different species, that is an unresolved trait. It's uninformative. You cannot identify the relationship between species A, B, C, and D. So our tree on the left, unresolved, we don't know their relationship. The tree on the right is the same four species, same exact arrangement, but in this way, we can resolve them because we have clear nodes that say that A and B are more closely related to each other, and C and D are more closely related to each other than either sets are to the other. So A and D are more closely related to each other than either is the C and D are more closely related to each other than either are to A and B. And this is resolved because we have clear nodes that describe this relationship. Make sense? So the issue with phylogenies is the number of alternative hypotheses. If we started this unit, we talked about traditionally we only have two hypotheses, which is stupid and ridiculous, but it tends to be what we do. We have the null, it's random, and we have the alternative. Well, this is the null, it's just random. This is one alternative. The thing about phylogenies is you have many, many different alternatives. If you remember when we started the class, when we had three species, we had three hypotheses. A and B are more closely related to each other than either are to C. B and C are more closely related to each other than either are to A. A and C are more closely related to each other than either are to B. Three hypotheses. But it is a factorial relationship. So as you increase the number of species, the number of hypotheses goes up dramatically. When you have 4 species, there are 15 different distinct alternative hypotheses. There's one null, unresolved, and then 15 alternative hypotheses. Well, you can use behavior to evaluate that. I mean, there's not a lot of behaviors, but there's clearly more than 15, so that would work. Especially if we start to subdivide them out, like we have grooming, a grooming antenna with mouth parts, grooming forelegs with mouth parts, grooming hind legs with mouth parts. That's 3 different hypotheses just for grooming. You can start to pull those out, and maybe that works. When you have 5 species, there are 105 alternative hypotheses. That starts to go beyond what behavior can do. There's just not enough behaviors, derivatives of behavior. When you get 6 species, you have almost 1,000 alternative hypotheses. Equally valid hypotheses before you use data. When you get 7 species, it's 10,000. 10,000 alternative hypotheses to describe the relationship between 7 species. How many species are here? We have 3 that are the outgroup. How many are here? Guesstimate. Guess. I don't care what the right answer is. 50, 60, does that seem reasonable? How many alternative hypotheses are there? At a minimum, millions. Realistically, more like billions. Alternative, equally valid hypotheses. At this point, we can't even use standard procedures. We've got to use heuristic and bootstrapping procedures to start to generate a tree, and you need stupid amount of data to discriminate between one group and another. Well, what's the best trait for discriminating between one group and another? Because there are stupid amounts of data. What's the most common trait? By a long shot. Most common trait in biology. Oh, this is great. Let's take a break. Come on back in 5 minutes. I will come back to that question. When someone is randomly up, it's going to get it first. And if you've already answered the question, you know it's not you. Enjoy the calm relaxation of your break. I'll see you at 9:22.
If you'd like to learn more about the world of history and the wonderful members of Lancashire, please feel free to subscribe to the channel. Thank you, sir.
Hey.
I have a question about the reading journal.
Oh, one second. Way too loud. Way too loud. Yeah, what's up, Audrey?
How can I do better? Because like I'm reading the rubric and like what I'm doing wrong.
You got a few minutes after class? Let's talk. Yeah.
Yeah, because then we can actually look at your submission and sort of talk about what's being looked for and there. Yeah, happily. Listen, thank you. We got. Adriana, you are here. Rachel, you are here.
Let's see. We're not getting audio. We're not getting audio. Not getting audio. Now we get audio there. Now do we get audio here? Now we do. Okay. So you need stupid amounts of data. Biologists are very valuable for understanding the evolutionary behavior. Behavior is not very valuable to developing a tree. I asked you why. We have the idea that they're complex phenotypes. They're made up of a lot of other traits, and there are relatively few behaviors. So you could use behavior for a tree of 3 different species, 4 different species, maybe even 5 different species. But as soon as we hit 6, not a chance. Unless we're doing something like the behavior we did over the weekend with that particular paper, which was really used to resolve. And they only had 42 characters, if I remember correctly, but that allowed them to discriminate between the 2 driving hypotheses of morphology and genetics. What is the most useful trait to build a tree? Because it's stupid common, like country mile. Audrey? And therefore, what is the most common trait in biology? Lewis, you want to go for it? Yeah, well, adaptations are sort of the result of things. And the adaptation paradigm argues that all traits are the result of natural selection, in which we have some, some ways in which we want to be a little bit more subtle about that hypothesis. Eating's a good behavior, but how many different forms of eating are there? Not many. I mean, normally you got an orifice, and somehow things get into the orifice. David, and then Hannah, and then Adriana. Mating's a behavior. How many different ways of mating are there? Well, you kind of do. There's external fertilization. There's internal fertilization. There's active mating, and there's passive mating. So things like grasses, kind of passive mating. Things like direct pollination, more active. Internal fertilization, and in animals, external fertilization, pretty active. But then sort of broadcast mating, where like corals and sponges and marine systems just sort of send out gametes, very passive. There's not many varieties there, kind of all the same type of thing. Most common trait, and it has nothing to do with behavior. Well, okay, I take that back. It's the foundation of behavior, but we do not consider it behavior normally. Even our units about it. DNA, by far. It's the most common thing, base pair sequences. Billions of base pair sequences. Each one of those base pairs is a different trait. There's only 4 types of them, don't get me wrong. But that variation between what's at the first base pair versus second base pair and so long along string. That's why we use genetics. Overwhelmingly use genetics. To a lesser degree, morphology, overwhelmingly use genetics. We don't use physiology for the same reason we don't use behavior. It's just not enough of it. It's not variable enough. It's too similar, too many similarities. Now, we've been talking about phylogenies, and we're talking about all the good things of phylogenies, but I'd be remiss if we don't also identify the fact that there are some problems. And there are problems with everything. There's no perfect thing in science. Science is the best of our knowledge by a long shot. There's no better way of understanding things outside of science. There isn't. But we always do want to recognize both the good and the bad. One thing is that phylogenetic study is only as good as the tree. That's what you read this past weekend. The genetic tree describing the relationship between the social insects was good. The morphological tree was good, but there was clearly a problem because they were unresolved. They did not agree. And so the tree is only as good, I'm sorry, the study is only as good as the tree. One of the things that's really important is that trees must be constructed of independent characters. So as an example, when we talk about DNA, a gene is less useful than a non-coding area of the genome. And the reason why the non-coding area of the genome is more useful to create a tree is because each base pair can vary independently. But in a gene, the sequence is normally constrained because of the adaptation to produce a particular protein. So if you get a mutation in your gene, is that normally a good thing or a bad thing? In a gene that codes for a protein, in you, is that a good thing or a bad thing? A mutation in your gene. It is a bad thing. So Marvel can suck it. They're wrong. Mutations are not good things. We don't want mutations. Now, on occasion, a mutation is a good thing, but more often than not, a mutation is a bad thing. What is a form of mutation that too many of us are going to have to deal with in our lifetime, or someone we love is going to have to deal with in our lifetime? Cancer. Cancer is mutation of a base pair sequence in a gene that codes for a protein. It doesn't make the person better. Rarely, hyper rarely. More often than not, it causes problems. Characters must be independent of each other. So the base pair sequence in a gene is not a good set of traits to use to construct a phylogeny, but the base pair sequence in a non-coding of DNA can be very valuable because each of those base pairs can vary because each of them can evolve without any effect on the organism's fitness. Those base pairs are neutral. There is a lack of data independence, where what you're trying to understand the tree is also how you're trying to define the tree. That's a problem. So you don't build a tree on behavior and then use that same tree to evaluate the evolution of the behavior. There is a lack of independence between the tree's construction and the use. It's one of the reasons why DNA is so valuable, especially non-coding areas, because they generally don't do anything. So then we can make the tree with a non-coding area, and then we can evaluate any variety of things. The genes that actually code for proteins, the morphology, the physiology, the immunology, the behavior. Behavioral characters, with the exception of this weekend's reading, are many times very hard to classify within a tree. That's fine. Parsimony, we talked about this. Parsimony is great, except when it doesn't work. We just talked about a p-value, and you guys made some really good statements about it. With a p-value of 0.05, there's a 5% chance of making a mistake. That's what that is literally telling you. There's a 5% chance of making a mistake if you reject the null. There's a 5% chance of making a mistake. So when you use a p-value of 0.05, how likely are you going to be correct? 95%. And what's the chances you're going to be wrong? 5%. That's science. We have the best of our knowledge. We don't have absolute knowledge. Parsimony is very good, except for those cases where it doesn't work. And the thing about parsimony is you won't know when it doesn't work until you get in more trees and more evaluation, maybe a different tree starts to refute. Data collection can be really difficult to construct a tree. What's the easiest mass data to collect today? DNA sequences. You get a DNA automator. It's when I was a kid, we didn't even know really how they operate. We were just doing gel electrophoresis of protein sizes. We didn't know what the heck was underneath. I mean, we knew there was DNA and that was all resolved. I'm not that old, but that was there. DNA automation came out during my lifetime. I remember the first time I saw a machine. It was sexy. That is now relatively easy to collect. You can just actually collect some tissue, send it out to the company, they'll process it, and it's not even that expensive. And you get the sequence of the information out. Very rapid order. It's brilliant. Other types of data are much harder to collect. Morphology is difficult to collect. You can't go out and automate it. You literally have to get it underneath a microscope or get it in front of a specimen and start to track it out. That is a difficult thing to do. And then last but not least, large data sets are needed. Large data sets, stupid large data sets. And actually, we've gotten to the point where you also need to model out. You can't actually use the data to come to absolute solutions. You have to model out your solutions through heuristic procedures. So these are all problems. They're survivable, but they're problems. Real quick things for the readings, since I mentioned it a few times right now. The readings are low stake grades. One, I like you to read. I like reading. You should like reading too. Two, I like you to think about what you're reading. These are what you're sort of giving me back. But the point of these assignments is sort of giving you some framework for what we're going to be looking at in the scientific biography, which is a very substantive project we'll do throughout the semester. And a few of the things that I'm looking for in these readings is you to give me some opinion, but really give me that opinion based on a good logic with good evidence. I'm not asking you to draw from different sources to derive that evidence. I am asking you to use what is in the reading for that evidence. So as you read these, I want to make sure that you're sort of picking up on those pieces. I'm giving you grades, I'm giving you sort of feedback. Don't hesitate to ask. If you're not seeing it, you're not getting it, do ask. The rubric is there to help you. That doesn't mean it's helpful. So if coming in sort of provides some additional information, we have office hours right after this class, use them. These are low stake assignments so that by the end of the semester, you have a really good sense for how to construct an idea out, how to explain an idea out, and how to use evidence to explain that idea out. And there are two major reasons why I want you to do that, actually three. I want you to learn how to ask questions, but we'll get to that later in the semester. I want you to learn how to use evidence to make a case for something. And if there's one weakness in scientists and science students that really hurts them in anything they want to do after graduation, we are terrible with explaining out our evidence. We are very good for stating our evidence. We state it, we assume we're done. We give you a p-value as an example, and we assume we're done. We write a statement, there's a citation at the end of it, and we assume we are done. Now, we don't assume we have to actually explain what is actually being stated. So as an example, for a little silly result sentence, I'll go back a few slides to make the point. A very simple sentence for here, where a lot of scientists would stop, is saying that there's no relationship between the striping pattern of the ungulates and the likelihood that they're in a light forest. Or maybe even better, I'll go back one more, so I think I make a better case for here. There is a relationship between spotting and ungulates and hiding. And then they stop. They give the evidence in the stats, but then they stop. Have they explained it? And I didn't say interpret it. Have they explained it? And the answer is no. You got to go to the next step. Those ungulates with spots tend to hide more than ungulates without spots. You got to actually add the second part in it. In scientists, we are infamous for incomplete statements, which actually hurts a lot of people when they try to get a job because they'll put in like a cover letter. That's just a few sentences and assume they're done. Oh, I got good grades. Oh, I'm good. Hire me. Take me on. Versus someone said, I got good grades because I've developed these skills, which allow me to sort of build out these ideas. I'll be able to apply them to what we're doing here based on your call and your conditions. That second person who actually puts things in context, the grades are good, but then gives you why they're good, and then how that's going to apply to a situation does much better than a person who says, oh, look, I got a GPA of 3.35. No. Yeah, sure, why not? That's good. Or 3.85. That's also good. Does that make sense? So that's what we're trying to do with these little writing journal assignments. And then those are all building up for the last part. Questions on that? All right. If you guys are good, I would like to introduce behavioral genetics. For the rest of the semester, we will be talking about mechanisms of behavior. So we have just this little brief introduction of behavioral ecology, and we use behavioral phylogenetics to sort of illustrate that process, where behavioral ecology is the evolution and adaptation of behavior. And again, it's better described as evolutionary behavior. Mechanisms of behavior, by contrast, are trying to understand the how and the when in terms of the individual organism's response. So we tend to look at things like the molecular and physiological basis of behavior. So we're going to spend the entire semester talking about the genetics of behavior, the endocrinology of behavior, the neurology of behavior. Basically, think of any class you have in biology, and we're going to add on 2 words of behavior. Make sense? So these are key information in terms of any behavioral study. Not only do you have to understand the adaptation and the evolutionary history, but you have to understand the how, the mechanistic why. And as a reminder, with that building ground squirrel and dispersal, there were a few things. One, there was an age issue. When the animals hit a certain age, the males hit a certain age, they were more likely to disperse than before that age, around 100 days, if I remember correctly. That's a win. That's a mechanism of behavior. Also, the how. Gonadal steroids were important. Fat bodies were important. When certain levels of gonadal steroids and fat bodies were reached, the animals were more likely to disperse. That doesn't tell you the why. Maybe it's incest avoidance, but it's absolutely key and critical to understand sort of the emergence of that behavior. And without it, we're missing out a lot of information, a lot of understanding in terms of the behavior. Now, I'll probably say it a few more times during the course of this semester, but I want to sort of reinforce it right now. All of these really get at sort of the holistic understanding. To understand sort of why some of these things occur, we really do want to understand that entire process. So with that nuptial gift in the scorpion flies, the balloon, which is so wicked cool, we want to get the mechanism of the female as well. What is she evaluating? What is she picking up on? What is she not picking up on? Are there certain genes which are downregulated, which no longer has her process out the item food that should be associated with that balloon? Are there other genes being upexpressed, which are actually evaluating what she's looking for in that balloon? These are all key to understanding the reasons why the balloon is being used as a nuptial gift. So, what can the information tell us about the evolution of dispersal, and why do we need to know these mechanisms? These will be key points we try to address during the rest of the semester. It's not just the mechanisms, but it's the mechanisms in context to other considerations. So let's start with this. Let me see if I can hide. Nope. Hide video. Nope. Hide. There we go. Can genes influence behavior? When and how can genetics influence behavior? So I'm going to leave this as a hanging question. And we're going to start with this on Thursday. But I will add, when I started my job, this led to some of the biggest fights I've ever seen in a class. I'll be interested to see what happens on Thursday, but I want to expose you to the question now so that you can think about it, because it's actually started to return as a politicized conversation. In fact, I think I told you guys about the dog interview of last week, right? Where that was the basis of the reason for the report. Am I talking? Don't pack. I'm giving you enough time. It's very disruptive and disconcerting and makes me anxious if you're packing. Are there any questions? We will be doing both the lab, but we'll start the class with a few things in terms of behavioral genetics, and then we'll switch over to lab on Thursday. Just so you know, next Thursday, we will be meeting virtually. I'll be at a conference up in Savannah, but I'll just stay in my hotel room so we can have class. And then after class, I'll go to the conference. Any questions with that? All right. Have a lovely day. I'll see you in 2 days. You as well.